And then our final talk of this session is from Jay Hickman from Hesperos, looking at human neuromuscular junction platform to evaluate botulinum toxin potency.
Thank you very much, Patsy. Thank you, Nick, for introducing the NMJ stuff. I knew you'd do that, so I wouldn't have to do it, so I didn't include it in my talk. I just want to say we have been just accepted into the ISTAND program for the replace the mouse lethality assay. So that just occurred recently. So I want to kind of give you an idea of what Hesperos does. I've worked in the area, what clinically relevant functional readouts for about 30 years now. So what does that mean? So you go to a doctor's office, they don't immediately stick a needle in your arm, be able to put out fluids, look at biomarkers, right? Look how you're walking, how you're talking. They might listen to your heart. They check your reflexes. They might ask you about elimination. What they're really checking on is your muscles contracting okay. Okay, is your cognitive function okay? Is your heart working well? Is your neuromuscular junction functioning well? Is your barrier integrity, things like GI tract and kidney working well? And so what we can do is take silicon devices, modify the surface of them, okay, so I can integrate cells. So I've got cardiac cells here. When it contracts, it deflects the cantilever, and then the laser moves, I can get force. We can pattern cardiomyocytes to be able to get conduction velocity, QT interval. Again, skeletal muscle force on cantilevers. Wide variety of different neuronal PNS and CNS model systems. We can still look at biomarkers, but we don't have to. But we're trying to do this all without cell death, okay? What we're really trying to do is have that one-to-one correspondence, what you see clinically, with what you're actually observing in our systems, okay? Now what we can do, we can then take all these systems, culture them out on their own chips, okay? Put them in, time it so they can put them all together into a base platform. We can put a gasket on top of it, so have a media then flow over top of all the different organ systems. It's the same serum-free media. I banned the use of serum in my lab 25 years ago, so everything is, the multi-organ system is in the same serum-free media. We can put a clear top on it, so we can do all the advanced imaging studies. So say if you've got an organoid you've been using for cardiac, we can then look and see what it does in the same media, in the same conditions as the functional readouts that we're doing. So one of these multi-organ systems we did is the neuromuscular junction, where we can actually have two different chambers. We have tunnels to be able to have the axons go through and then innervate the muscle. Nice thing about this is it's electrically and chemically isolated, so I can put a drug on this side and only see the effect on the motor neurons, or a drug on this side to see only effect on the muscle or the neuromuscular junction. And again, I can stimulate this side and be able to see what's going through the axons to the muscle, or then stimulate the muscle directly. We had published the first dose response curve for Botox back in 2018. And so obviously this has proceeded since then. And so this was the basis of our application into the looking at the botulinum toxin system. And so basically we have now utilized that system to be able to do dose response curves for 3 hours, okay, looking at frequency changes, okay, getting very good tight IC50s for each different frequency. The nice thing about looking at different frequencies is we can actually looking at mechanisms as well. So if we want to look and say, look at different serotypes in the future, we can use these different measures to be able to do that. We can also look at 24-hour, and again, looking very tight IC50s in terms of 24-hour collection. There's advantages going 3 hours versus 24 hours. Again, mechanistically, different things are happening with the Botox, okay, depending upon what the time frame is. And we can also do 72 hours, which is about the time frame for the mass lethality assay. So again, the things you can see, and again, getting very good dose response curves, okay, and the EC50s that are all pretty comparable, okay, with those different model systems, okay. We can also now, this is create a high throughput system. Our lead engineer, engineering director, Chris Long, is here today, has worked on this system. Okay, we can now do a high throughput NMJ model system in the lab. And this is the data we can get for looking at NMJ number, okay, looking at different frequencies. And again, getting very, very good correlation between the EC50s we're getting for these. So this is the data that was submitted as part of the letter of intent, okay, that has now been accepted. We're now going to work with the ISTAND program to be able to get a qualification package together and then expand that out into other different things in terms of food, different serotypes, okay, as well. Now, one of the things that was asked for in the proposal we put in was, can you also take this neuromuscular junction system and do other things with it, these disease aspects and things? I'm not going to get into all the data because I only got 10 minutes, but I just show you that these are the approved, where we basically repurpose drugs into INDs, okay, that enabled phase 2 clinical trials. CMT with Vanda Pharmaceuticals, we did precision medicine for a single rare disease patient. They're going to be presenting the clinical data for that next week at the muscular dystrophy meeting that will be in Orlando. And basically, I know that the patient is actually getting better at this point with that treatment, okay, which is a first for that disease. We've also done ALS with Apellis, Argenx ALS, myasthenia gravis with Dianthus. This phase 2 has now been successful. They're in their phase 3. We can also modify the system and also make it a conduction velocity model system, be able to look at peripheral neuropathies, and say also for Dianthus, we also did multifocal neuropathy and CIDP. And then initially with Sanofi, we also did CIDP. This phase 2 has been successful now in a 2-arm phase 3 as well. So showing that we can actually have not only utilize these systems for safety aspects, but also utilize it for predicting efficacy for repurposed drugs. Now, this is a new finding that we just had, is that I've always been curious in terms of, Alzheimer's disease, the fact that people also get motion problems, and you look at this in terms of gait, you look at this muscle mass, gait speed and everything. There's big differences, okay, between with male and females with AD in terms of your peripheral functions. It's not just a CNS disease. So what we did is we actually then use that 2 different mutations, the presilin-1 for Alzheimer's and the APP mutation. And then we had the isogenic, the wild type and the isogenic APP. Okay, and you basically see here for the 2 normal, you don't see really any effects in terms of fidelity. And again, these are the traces Nick had shown you before, with the mutations, it goes down. Okay, you basically still see with the mutations we're getting, fidelity is going down for these different mutations. Okay, again, you see this also in terms of the higher frequencies going down much more for the presilin-1 than it is for the APP, which is really interesting because in the CNS, presilin-1 is hyperexcitable. For the motor neurons, it actually has a larger deficit. So it's actually doing different things between the peripheral nervous system in terms of the AD mutations versus the CNS. Okay, and lastly, you can actually then see, again, in terms of fatigue index, up makes it worse. You can basically see the fatigue index is going up in terms of the 2 controls. This was basically just accepted in IMPRESS in Alzheimer's and dementia. And one of the reviewers said this is the first demonstration showing that Alzheimer's disease mutations, okay, can actually affect directly the PNS. The deficits are not all coming from the CNS, okay, and that will be published in Alzheimer's and dementia in the next couple of weeks. Then lastly, I wanted to talk about, we can also do digital twins with these model systems. We've just recently had a publication for a malaria on a chip digital twin, where the Gates Foundation asked us to create a liver, splenocyte, endothelial cell model system, and then we'll be able to put the blood in there to be able to look at the parasitemia in that model system. And by looking at these different parameters, you'll be at the active concentration, and we're able to predict the PK, okay, for this in the model system, okay, in vitro. We then get the pharmacodynamics because we have the clinically relevant functional readouts, which allows us to predict maximum tolerated dose, no adverse effect dose, and EC50, which was in good agreement with what you see in vivo. And then actually these were acute dosing, and then we can actually then plug it into the digital twin. This is what the acute dose would show, but then we can actually then model what the actual dosing was and get the exact parasitemia. This was published in Advanced Science. So we can go through these systems looking at safety. We can also go these things looking at efficacy. We can build these model systems, and then we can then predict in vivo outcomes by collecting the PD and PK in these model systems. And the key thing about all of this also is these are all commercially available services right now. Okay, this is not something in the future. These are all available right now for the model systems. So that, and then that's kind of what I want to show you today, that we can kind of go through very basic ideas, very mechanistic, all the way through to, doing clinical data in, submissions for INDs in collaboration with pharma folks. Thank you.